CRISPR just got a new cousin, and it's a wild one.
Feng Z's lab at the Broad Institute just discovered TIGR, a new class of CRISPR-like effector that provides a compact alternative to conventional CRISPR, and it has a badass name.
Feng Z and his lab are among the pioneers of CRISPR, having been one of the first labs to describe the CRISPR system. Also, a bit more than a year ago, they reported the discovery of Casts, or CRISPR-associated transposases.
Even more striking was their first report of a new class of eukaryotic RNA endonucleases, called Feng Z, in 2023.
At this point, you would think that they couldn't possibly discover anything else, right? Well, you would be wrong.
As a matter of fact, last week, Zhang's group published on Science and discovered a new class of CRISPR-like effector called Tandem Interspaced guideRNA system, or TIGR.
This new class of nucleases are mostly found in bacteriophages, archaeoviruses, or parasitic bacteria. And the question is, how the hell did they find them?
We'll say it in advance, the research was complex, but we'll try our best to summarize it for you as smoothly as possible. Just bear with us.
First of all, the team used various databases to look for proteins with an RNA binding domain similar to that of SPCas9.
This way they tried to find unknown proteins with the potential ability to bind a guideRNA or similar molecules.
Among the hits of the research, they could find recently described guideRNA transposases, the bridge RNA system which we also recently covered. That was a sign that they were looking in the right place.
Following this strategy, they identified a new domain that resembles the RNA binding domain of both SPCas9 and the bridge RNA transposase, the NOP domain.
Using NOP as a proxy, they dug further and discovered that in some bacterial genomes, NOP is located close to arrays of repeated sequences.
This is as difficult as finding a golden nugget in a pile of coal, but they managed.
These repeats they found are similar to those of CRISPR, but with a different structure. Instead of the same repeat separating each spacer, the new system has a couple of recurring repeats interspaced by a unique sequence.
For this reason, they dubbed it Tandem Interspaced guideRNA system, or TIGR.
The full TIGR system is composed of a TIGR-associated protein, TAS-A, which contains the NOP domain that we mentioned before, and two other TAS, called TAS-H and TAS-R, which may contain catalytic domains similar to SPCas9.
These three proteins are required to process the TIGR-RNA, the TIGR version of a guideRNA, which defines the target.
However, we have two main differences with the CRISPR system. First, there's no PAM required. And second, the guideRNA is not made of one single spacer but two, and both spacers, A and B, specify the target, one on each strand.
After characterizing the TIGR system, they tried to use it to edit the DNA in human cells. And while the editing efficiency was low, they could still show that, with further optimization, TIGR may be translated to human cells.
Overall, this amazing work shows how CRISPR and CRISPR-like systems never cease to surprise us. TIGR represents a whole new family of RNA-guided nucleases in prokaryotes and their viruses.
While this system is brand new and a lot needs still to be understood, Feng Z and his lab could already show its potential as a genome editor tool in human cells, a promising sign for its future.
We here with the CRISPR will follow closely this technology and its development and be ready to tell you all about it.